So again, along the theme of biomarkers, at this time, let's talk about what we can find these microbiomes in your urine. These are my disclosures, and we have um, the adult human body contains about 30 trillion human cells, and then 39 trillion microbial cells. And same thing when it comes to the genes. The microbial genes are much larger percentage than the human genes. These microbial genes are responsible for initiation, progression of certain diseases, and it also provides some protection against diseases. Many other beneficial functions, digestion of food, breaking down lipids, etc. So we are particularly interested in this urogenital microbiota, which is a community of microorganisms that lives and resides in the urinary and genital tract. So the next-gen sequencing enabled us to get a good understanding about these profiles, microbial profiles. And one of the things now we find is, the old school says urine is sterile, but no longer it is sterile. And the urinary microbiota shares many species with the gut and vaginal microbiota. And understanding the normal, I put that word in quotes, so the normal microbiota in healthy individuals might give us an idea when we are fighting like UTI and other diseases, what are the organisms that are responsible. So how do you characterize these microbiomes? One is using community state types, which are either defined by one dominant species or a combination of species. For an example, if you look at vaginal microbiota, it is characterized by 5 different community state types. Type 1, it's Lactobacillus crispatus, and you can see then there are several Lactobacillus family members in there. But if you look at type 4, that is a much more diverse group. And why Lactobacillus? It keeps the vaginal pH, between 3.8 more acidic, and produce lactic acid, and keep the genital tract inflammation down. So there are many benefits for these things. And we did a study, and the objective of this study is to find the urinary microbiomes in healthy men and women without urinary diseases, that is UTI, pelvic pain, urologic cancers, and not currently taking antibiotics. And again, as the characterization provide the foundation to understanding any departures from healthy conditions. It is IRB approved, so we consented symptomatic subjects were invited by various electronic advertisement notice to provide urine samples for us without any age restrictions, and we also provide a survey questionnaire that they need to complete. We collected midstream urine specimens, and the urine analysis excluded if it was positive for nitrates and leukocytes. And these were processed by Microgen DX in their Lubbock, Texas laboratory, and followed by next-gen sequencing, bioinformatics, and statistical analysis. Here is the patient demographic data, the equal number of male and female approximately, and the median age is 39, and the age range from 4 years to 85. And I've given you the distribution of the age here. Here is the questionnaire. Most of the women were premenopausal states, and about a third of our group were on antibiotics use, in the past 3 months. They said yes. And also about 10% of them were taking urinary medications, and then had more women had UTI in the past 3 months. And so let's see if these factors are contributing to the microbial profile. Here are the results to analyze. There were 609 species, and mostly dominated by 4 major phyla, Firmicutes, Protobacteria, etc. And the 19 most abundant species are given in this one. It includes Lactobacillus family, Streptococcus, etc. But let me move on to the more complete analysis to give you the details. Between male and female, there were 23 species from 5 genera that were significantly different. And these are all outlined here. You don't have to go through everything. But Lactobacillus crispatus and Lactobacillus densitani were the more abundant in females. This Streptococcus mitis was more prominent in the males. And the Lactobacillus crispatus has the largest mean increase in females relative to the males. So I have sort of highlighted these things here on this side. And that is between the different genders. Now, the community state type analysis, men, of course, can be clustered to a single community state type. Everybody falls into the same thing. Of course, the women, we were able to classify them into three groups, three community state types, and the memberships. And the principal component analysis here shows that the axis 1 and axis 2 were significantly different and explain about 45% of the total variation in females. And the age of the females were non-randomly distributed in the community states, especially CST1 had a more older population compared to community state type 2, and that's a significant. So the age also begin to play a role in how you classify these things. And here are the significant species, because the previous one you could not tell what are the dominant types in each community state types. You had to do a separate statistical analysis for that. And when you do that, pairwise comparisons of all 23 species, the charts show highlighted ones, and E. coli is probably the most present. And so consequently, the state type 1 has E. coli, Enterococcus facilis, and Lactobacillus inus, those things, and then community state type 2 dominated by Lactobacillus crispatus. And so each one of these state types will have a group of dominant species that describe these things. Now, some other interesting observations. We had the question here. We tried to relate the questions and answers to these community state types, especially in females, because the men all clustered into one community state type. Pelvic pain in the last 3 months, yes or no, was associated with the classification of third community state type having the lowest percentage of yes, and it is statistically significant. The other two results are not statistically significant, but it is sort of heading in that way. If you say currently taking urinary medication, yes or no, had a notable association with the community state type, with none of the yes responded were in the third community state type, which is dominated by, I think, E. coli. And the menopause pre-post also had a notable association with the community state types. 11 of 16, almost 70%, post-responded were assigned to first community state type 1. So as you can see, if you are trying to understand the healthy, microbiome profiles of individuals, it has a lot to do with the male versus female, and then, some of the, other factors, such as whether they had, pelvic pain, current medications, menopausal status, etc., that also need to take into consideration. So, on conclusions, urinary microbiome from healthy subject can be organized into community state types subject to patient variables, gender, age, pelvic pain, etc. Gender and age explain the compositional differences and species abundance. So the interpretation of next-gen sequence analysis of the urine specimens in you are diagnosing UTI or any other disease, possible bacterial infections, as a cause of bladder symptoms and pain, we need to consider the normal community states with other patient variables. So it is a good take-home message would be it is not one size fits all, but there are many other factors you need to take into considerations. And thank you very much. And the work was done by everybody here supported by Microgen DX and Dr. Crawford, Dr. Nichols, and all of them contributed to this work. Thank you very much.